So Mendelian extensions 2 in what we'll be really discussing today is scenarios in which we can start seeing deviations from expected Mendelian patterns, right? And I'll say these now, I'll say it again later. It's not like we are deviating from Mendelian genetics. It's just that the genes that we're looking at behave in such ways that alter the phenotypes that we observe. But the genes are still behaving the way that Mendel predicted or established or modeled, okay? It's just that the phenotypes are highly influenced by how the particular loci that we're looking at interact with one another. Now we know a little better on a molecular basis, and we'll touch on that as well. Okay, so we left it here. We said that anytime that we have a cross that involves several loci, in this case 5 of them, and we're asked the probability of a particular outcome, a particular genotype, one convenient way to approach this is to split up the cross into a number of monolocus crosses that is equal to the number of loci that you had originally. So if the cross involved 5 loci, we'll split it up into 5 individual crosses, each of them dealing with each of these loci, one at a time, right? And that way we can simply calculate the probability of each of the genotypes for each locus and then multiply across to obtain the combined probability of the desired genotypes, okay, or allelic combinations. That's where we left it the other day. Are there any questions about this? The only thing that I'll say again, I kind of mentioned it in passing on Tuesday. When you're thinking about splitting these crosses into individual monolocus crosses, bear in mind that not all of them are going to be monohybrid crosses. I don't want to make it sound like now everything turns into a monohybrid cross with 2 heterozygote parents. So here we have an example, starting with the M locus, where that's not a monohybrid cross. One of them is heterozygote, but the other one is a homozygote recessive. If anything, you might argue it's a test cross, right? And this is another interesting example. We have a cross between a homozygote recessive and a homozygote dominant. So it's not as simple as remembering the 1 to 2 to 1 ratio of genotypic outcomes, because that 1 to 2 to 1 of homozygote dominant to heterozygotes to homozygote recessives applies only to the monohybrid cross. You'll have to treat each of these crosses separately, if you will, and in many cases you may have to reason through the cross itself to try and calculate what the probability is. So for instance, for that homozygote recessive T to a homozygote dominant T, the probability of a heterozygote happens to be 1. The first parent is contributing always a dominant, the second parent always a recessive, so actually all of the progeny would be heterozygote. Out of this, the probability of the first parent contributing dominant and the second parent contributing recessive is 0 times 0. But when we're asking the probability of a heterozygote in the progeny, we don't care which way it is. And that's why we need to consider both possibilities. And it turns out that order, with the first parent contributing the dominant and the second parent contributing the recessive, is indeed 0. The probability of that is indeed 0. But the probability of the first parent contributing a recessive and the second one a dominant is a flat out 1. And then we add them together because either alternative works, and we use the rule of sum, and we get 1 plus 0, 1. Order doesn't ever matter. The point is that when we're asking about a genotype, what we care about is the combination of alleles, not so much which parent these alleles came from. The only time it matters is when we're trying to calculate some of these things, and we need to remember that for an individual in progeny to be heterozygote, it doesn't matter if they inherit the dominant from mom and the recessive from dad, or dominant from the ovule and the recessive from the pollen, or vice versa. We good? Can't think of anything else to stress about this. Okay. What about if we want to use a Punnett square to solve some of these multi-loci crosses? So the rules for solving these crosses with a Punnett square are not different from the monolocus crosses, we'll first want to determine the number of rows and columns. And again, we'll use that 1-2 rule that I mentioned before. So what you do is, first you go parent by parent. So we'll start with the first parent here. It's a double hybrid. And we ask ourselves, how many different allele types are there for the first locus, for the R locus? Allele types, how many are there? 2. Write down a 2. How many allele types for the second locus? 2 as well. So you multiply those, and now you determine that you need 4 rows in this Punnett square. We do the same for the columns. How many allele types for the first locus in the second parent? 2, and 2 types for the second locus. 2 times 2, 4 columns. As you will see, this exercise becomes really, really handy when you're dealing with more than 3 or more loci. And we have a slide dealing with that. So first you establish the number of rows and columns. And then comes what is arguably the trickiest step in this process. And that is labeling the rows and columns. And when I first put together this slide, I could think of a very common mistake. It turns out there is 2 very common mistakes, and we'll address them. Right? So the first very common mistake is to say, okay, I just need to distribute the alleles across these rows and columns. So I'll start with the columns. My first column will be dominant R. My second column, recessive R. Third column, dominant Y. Fourth column, recessive Y. Why is that a problem? So you are, in a way, what is it that we're labeling when we're labeling columns and rows in these Punnett squares? What is it that we're describing? Are we describing the genotypes of the parents? The gametes, that's right. What we're trying to figure out with the columns and rows are the types of gametes that these, in this case, plants are producing. And the problem with doing this, common mistake number 1, is that this implies that the gametes produced by this plant will contain either alleles for R or alleles for Y. But we know that gametes should have 1 allele for every locus. These plants are putting either allele for each locus into gametes. And this makes it sound like this particular, let's say, pollen grain carries the dominant for R and nothing else. This pollen grain carries a recessive for R and nothing else. And that's not the case. So when we split the alleles across columns and rows, we need to be careful. We need to remember that there should be an allele, either allele, for each locus in each gamete. So what is common mistake number 2? Common mistake number 2 is to say, okay, now I got it. There has to be an R and a Y. I'll figure out whether it's capital or lowercase later. So what I'll do is I'll say, okay, I'll do it with the rows. I know that half the gametes will have dominant R and half the gametes will have recessive R. And I'll do it again, but for the Y locus, half my gametes will have dominant Y, half my gametes will have recessive Y. Why is that a problem? Exactly. The one thing that Mendel's second law established is that alleles for each locus will be distributed across gametes independently from what happens in other loci. So the dominants for Y don't always have to go with the dominants for R, and the recessives for Y don't have to always go with the recessive for R. But it's a very human nature tendency to say, I'll go in order, first the dominants, then the recessives, and then you end up with this that implies that all of the gametes that these plants produce are of 2 kinds, either 2 dominants or 2 recessives. When in reality we know there should be 4 kinds with equal probabilities, with a dominant of one, a recessive of the other, vice versa, or both dominants, both recessives. So how do we get to that? Well, so one way to do that, and to each their own, but what I suggest is you go letter by letter, right? And you start dividing it such that for the first letter you do half and half, but then for the second letter you go 1, 1, 1, 1. Did that make sense? So for the first letter, you split it half and half. And for the other one, you alternate. So now we ended up with all possible, real possible combinations of alleles in those gametes. And because both parents are dihybrid, this is a true dihybrid cross, then we know that in the columns is, in the rows is going to be the same thing. So I'm going to erase some of this. And let's do it again. So what I would do for the rows is top half dominants, bottom half recessives, and then for the second locus, I would alternate. So now that we labeled, we can solve the cells for genotypes and phenotypes. And what we do is, as usual, we write the letters that we see in a corresponding column header and a corresponding row label into that cell. So for that first cell over here, that would be dominant R, dominant Y, dominant Y. I pick any square here, like say that one over there. So that would be recessive, recessive, dominant, dominant, and so forth. So you solve the entire square. This part can be a little time consuming, and it can, I'll be honest with you, it can be a little error prone. So you have to be careful when you do this. And that's only filling out the square for genotypes. Then you have to, in addition, figure out the phenotypes that correspond to each of these. There's no rule for this. It's very personal. You can use letters to indicate the phenotypes. So one thing that you could do is you go in and then you say, okay, this is going to be smooth and yellow, right? Let me find one that makes sense. This guy here would be RR, little y, no. This guy here, RR, little y. So that one will be smooth because it's heterozygote for the R locus, but green, okay? Again, a little time consuming, but the reason why doing this with Punnett squares pays off is that then you have all the information in one place, and all you need to do is go around the square circling whatever question you're being asked. So if the question is, how many plants in the progeny will produce green peas? So then you're going around the square looking for any cells that have a G in them for green. How many plants produce smooth peas that are yellow? Then you go around the square looking for S, I suppose, for smooth or round, and Y for yellow. Yeah? It takes a little practice, but once you get the mechanics, it's not that bad. Okay? And also remember that the actual proportions that you're looking for are the number of cells that show what you're being asked. Let's say your cells of interest, in the sense that those are the cells that you're interested in, divided by total number of cells. In a full dihybrid cross, that total number of cells will be 16. And if we were to really solve it, and that's shown in the next slide, if we were to really solve it, we would be able to see the 9 to 3 to 3 to 1 distribution of phenotypes. Over the total number of cells that you have in that square. Why am I not saying 16? Why am I not saying number of cells divided by 16? Because there could be more than 16 if you have more loci, but let's say that you're looking at a dihybrid cross. The most varied that you can get is 16, but you could get less varied than 16, right? One of the parents could be homozygote for either locus, and that will reduce your square, right? So in that case, very careful. Don't divide by 16 by default or as a reflex, because it's really down to how many cells you have in your square. So it's the total number of cells, or the number of cells that you're interested in, divided by the total number of cells. Now, if we were to apply Punnett squares to the cross that we were dealing with before, right? One of the things that you may encounter out there in literature, in websites, papers, books, is that for one locus, your Punnett square should be a 2 by 2. For two loci, a 4 by 4, which is what we just did. For three loci, an 8 by 8, and so forth. For a penti-loci cross, like the one shown up there, should be 2 to the 5th times 2 to the 5th. And that is 32 times 32. That's not easy to draw, right? It would look something like this. Not fun. Not fun. So when you're approaching a multi-loci cross and you're deciding to see if you would like to apply probabilities and split it into several monolocus crosses, or you might want to go ahead and draw the Punnett square, one of the first things that you need to decide is how much you can reduce this square. Because the 32 by 32 is without any type of reduction. And we've seen before that when organisms are homozygote for one or more loci, even though they're producing twice the number of gametes with a particular allele in them, it's only one type of gamete carrying that allele, if that makes sense, right? So an organism that is heterozygote for the locus R, so it's dominant R, recessive R, will be producing two types of gametes. Half the gametes will have the dominant, half the gametes will have the recessives. A homozygote organism for the locus R will be producing only one type of gamete. So when it comes to that locus, two columns become one, two rows become one. That's what I mean by reducing the square. So let's do it. Let's apply that 1-2 rule that we talked about before. For the first parent, how many, let's say that we use the first parent for rows. How many rows due to the first locus? 2. How many rows due to the P locus? I hear some 2, some 1. You got to ask yourself, how many different allele types do I see? And in P, we see only one. We see only the dominant. How many for Q? 1. How many for T? 2. How many for S? 2. We multiply across, and this is 2 times 1, 2 times 1, 2 times 2, 4 times 2, 8. Yes? Let's go for the second parent. First locus, M. How many allele types? 1. P. Q. T. S. Total? 4. So 4 columns. So what appeared at first to be a Neimer, a 32 by 32 square with over 900 cells inside, turns into something that is a little more manageable. If you're playing music in the background, maybe having a drink, for those of you who can drink, and you turn it into an 8 rows by 4 column square. I'm not going to bother with the labeling of the columns, but let's see how we would approach labeling the rows, because that can be a nice exercise on how you do these things. So going by what I was saying before, how would you start with, so let me put it this way. What are the loci for which it will always be the same letter across? And we're talking this first parent here, right? So what would be the letters that are the same across all rows? P and Q, because the first parent is homozygote dominant for P, so the only thing that first parent can put into the gametes is a dominant P. And Q, because this first parent is homozygote recessive for Q, so the only thing that it can contribute is a recessive Q. So we'll do that. We'll go ahead and write that. So we'll do PQ. Cool. We took care of the easiest ones. What about the M? The M locus is heterozygote, so half the gametes will get the dominant, half of them will get the recessive. And I personally prefer to start broad, right? So I'm going to do top half dominant M, bottom half recessive M. So I will go in and do capital M, capital M, capital M, capital M. That's half the square. Recessive M, recessive M, recessive M, recessive M. For the T, here's where you need to be a little careful. Instead of alternating 1 and 1, 1 and 1, 1 and 1, my recommendation would be alternate 2 and 2, 2 and 2, 2 and 2. That looks something like this. T, T, lowercase, lowercase. Uppercase, uppercase, lowercase, lowercase. And then the very last one, the, what was it, an S? Yeah. The S. Now we alternate 1 and 1, 1 and 1, 1 and 1. So we do capital, lowercase, capital, lowercase, capital, lowercase, capital, lowercase. Whatever the question was, you would look around the cells. Filling up the square will take a little longer. I'll give you that. But once you did that, you look around the square looking for the cells that are of interest to you. And now your total is not going to be just a default 16 or a default 64. It'll simply be the total number of cells that exist in this reduced square, which would be 32. Don't panic. You will not come across anything like this in the exam, mostly because of time. There's no time to do this. But you might come across something like this, particularly if you engage with some of the problems from books, or if you go around online looking for genetic problems to practice, you may start bumping into some things like this. Okay? So now we're going to switch gears, and we're going to start talking about those true Mendelian extensions that that I've been sort of announcing for the last 2 lectures, right? And again, I said this before, but I'll repeat it. You may encounter some sources that refer to these things as Mendelian exceptions or Mendelian deviations. And once again, yes, we will talk about deviations from the classic phenotypic distribution of a dihybrid cross. That's fine. We will use that language. But we're not deviating from Mendelian rules of inheritance. These things are still being segregated, or alleles are still being segregated equally into gametes, and for the most part, they're going to be segregated independently from what happens to other loci. Next week, we'll deal with what happens when they do not segregate independently, okay? All right, so we said a number of times now that if you have a dihybrid cross, a cross between 2 individuals who are heterozygotes for 2 loci, what you can expect is that 9-3-3-1 phenotypic distribution with, for every 9 individuals, and these are relative proportions, right? But for every 9 individuals showing the double dominant trait, you'll see 3 individuals that show 1 dominant, 1 recessive, 3 individuals that show recessive for the first, dominant for the second, and only 1 individual that shows both recessives. Now, there are some conditions for that to happen. The first one is that there is full dominance, penetrance, and expressivity. That's what I like. You'll see in some of my questions and whatnot. I like to refer to those loci as totally dope, and they're easy to work with. They're no laughter? Okay. Sometimes you bomb. So totally dope loci are those that show full dominance, right? Penetrance and expressivity. And that matters because it means that when you have those conditions met, there is a distinct and clear-cut mapping between genotype and phenotype. What I mean by that is you know what to expect from a homozygote dominant, a heterozygote, and a homozygote recessive when it comes to phenotype. If there is full dominance, you know that the homozygote dominant and the heterozygote will look one way, and the homozygote recessive will look another way. Why is that? Because there is full dominance, right? Now, if you don't have full dominance, then we start getting into this terrain of, well, the heterozygote might look somewhere in between each homozygote, right? So if there is incomplete dominance and your dominant is red, your recessive is white, your incompletely dominant or your heterozygote might be a pink of some sort. If there is codominance, both of them are going to show up simultaneously. A little weird, but it happens, and so forth. So with full dominance is that we can trace this very clear, very distinct relationship between genotype and phenotype. That kind of goes away as well if there is incomplete penetrance. When there is incomplete penetrance, you expect a certain number of individuals with the dominant phenotype, and then it turns out that fewer individuals than those that you expected show that dominant phenotype. That's the definition of incomplete penetrance. And if there is variable expressivity, even worse, because now you have a range of expressions of a given phenotype, and it's very difficult to tie that up to a particular genotype. So we can predict a 9331 with dope loci. The other assumption that we're making is that characters segregate independently from each other. In other words, they follow Mendel's second law. And that's why when these dihybrid parents are producing gametes, we can expect that those gametes are going to contain all possible combinations of alleles in equal ratios. That's the very definition of Mendel's second law. And that expectation of 4 different types of gametes from each parent is what gives rise to this universe of 16 different allelic combinations that will turn, thanks to the loci being dope, will turn into the 9331 distribution of phenotypes. Okay? We didn't really hammer on this all that much, but just like we can come up with the 9331 phenotypic distribution for dihybrid crosses between dope loci, we can also come out with that 1 to 2 to 1 to 2 to 4 to 2 to genotypic distribution. We showed it briefly. We're not going to go back to it. It's too scary, right? But it's there. And you would see it in the Punnett square as well. Okay? So the bottom line is that all of this, all of this ability of predicting a 9 to 3 to 3 to 1 really comes from the assumption that parents are placing alleles for these 2 loci into gametes independently from each other. That what happens to alleles from one locus will not affect what's happening to alleles of the second one. Okay? And finally, and this is what we'll discuss the rest of today, there is the assumption that characters do not interfere with one another. The example that we've been talking about so far, these 2 traits that Mendel worked with, or these 2 characters that Mendel worked with, P skin and P color, did not affect one another, right? So the presence of wrinkle skin didn't have any effect on whether a P should be yellow or green. There were wrinkled green Ps, and there were also wrinkled yellow Ps. The fact that you saw more of the wrinkled yellow than wrinkled green, it's an indication that yellow dominates over green. That's it. It's not telling you anything about wrinkle affecting the way that P color manifests itself. Did that make sense? So there's no interaction whatsoever between these loci. So that's another assumption that we need to make, or that's another assumption, I should say, that is underlying the expected 9331 phenotypic distribution. But that we like it and that Mendel described it doesn't mean that that's the way biology works, right? So there will be many cases in which loci do interfere with one another. And in fact, that interference with one another can, I dare say always, we may not know the details, but it's explainable in molecular terms. What is that these genes are doing inside of cells, right? So let's go through a few examples of when characters do interfere with each other. So what happens when characters do interfere with one another? Let's start with this textbook example of what is called a simple recessive epistasis. And I'm going to start throwing some terms at you that you may have encountered before, but in case you didn't, we'll explain them in detail. So let's start with epistasis. Epistasis is the technical term for this phenomenon by which certain loci interact with others or interfere with others. A more human-centric way of seeing this is thinking about interference, because as we will see, the genotype of one locus sort of interferes with our ability of seeing the phenotype associated with a different locus. In more technical genetics terminology, we talk about genetic interactions. So we say that epistasis is simply when loci interact with one another. Now, epistasis, the root of the word, epi means on top, and stasis means to be or to stay, or that's where, I think it's probably Latin. So epistasis means ultimately to be on top. And the idea here is that when you have loci that interact with one another, there's one that is going to be on top of the other. And by being on top, I can't emphasize this enough, I'm not necessarily saying dominate over the other in the way that alleles for a given locus dominate over one another. It's more like what happens in one locus kind of dictates whether you can see what happens in a different locus or not. So let's go through this example. It's the example of simple recessive epistasis. And the way that it goes from a genetics perspective is that if of your two interacting loci, one is homozygous recessive, now you won't be able to see what's the state or the genotype of the other. That's why we call it simple recessive epistasis. When one of those two loci is homozygous recessive, one, simple, it's masking, it's covering, it's blocking our ability to see what happens with the other. So the classical textbook example that it's everywhere is fur color in Labrador dogs. It turns out that there is a locus that we call B locus that produces or codes for an enzyme that can turn dopachrome, which is a chocolate pigment, chocolate color pigment, into eumelanin, which is a much darker black pigment. So when the B locus is functioning, when there is either a homozygote working B locus or a heterozygote locus, the B locus happens to be haplosufficient, so only one copy will work, and it's therefore fully dominant, then you see production of eumelanin, and these dogs will be producing the black pigment. Okay? Only if they are homozygous recessive for the B locus would you see that there is no production of eumelanin, and these dogs are full of the chocolate pigment. So far, so good? So there is this locus, the B locus, that if homozygous recessive, it's chocolate. If heterozygote or homozygote dominant, black pigment production. Now the thing is that there is this other locus that we call the E locus, the epistatic locus, that when homozygous recessive, it will mask for us our ability to see either black or chocolate. Why? Because the E locus is in charge of depositing that pigment on the skin cells, on the melanocytes that are closest to the skin surface. Those are the ones that will ultimately color the fur of these dogs. So when the E locus is functioning, because it is homozygote wild type working or heterozygote, then we see pigment deposition. Whatever pigment that dog had. If it produced black pigment, we'll see black pigmentation. If it produced chocolate pigment, we'll see chocolate pigmentation, and we'll have one of these chocolate Labradors. But when the E locus is homozygous recessive, it's not working, the pigments are being produced, but they're not being presented on the surface of the epidermis of these dogs. They're not being presented where we can see them. And the dog ends up being one of these yellow Labradors. Did that make sense? So it is a case of epistasis because one of the loci, the epistatic locus, the E locus in this particular example, when homozygous recessive, masks the other. It blocks our ability to see what the state of the other is. So here you see the Punnett square that shows this in action. Instead of having the 9 to 3 to 3 to 1, 4 different phenotypic classes, you actually have only 3 phenotypic classes, right? You have either black dogs, chocolate dogs, or yellow labs. That's it. That's all you have. And the ratios change. And the reason for that is, in all of these cases of epistasis, you can always work it out such that you figure out which of these combinations of dominant-dominant, dominant-recessive, recessive-dominant, or recessive-recessive is masking or is being masked, and then you sort of collapse those together. I'll show you. So in this particular example, ideally, we would have expected a 9-3-3-1 of 2 dominant traits, first dominant, second recessive, first recessive, second dominant, and then the 2 recessives. But what's happening here is that the second recessive is masking the expression of the dominant. It's blocking it. It's covering it for us. And now these 2 collapse into 1. The presence of that recessive does not allow us to see the other dominant trait, as we would have expected from loci that do not interact with one another. So the epistatic locus, the E locus, is masking the genotype of the D locus, and therefore the phenotype of the D locus, when that epistatic locus is recessive. That is, of course, because this is a simple recessive epistasis. We'll see there is other classes. Here's the reason why I emphasize that. Every semester, I get this question. There's 4 types of epistasis that we'll cover today, and each of those 4 classes will come with a different deviation from these phenotypic ratios. So every semester, I get students asking me, do I need to remember all 4? That's your level of comfort. If you can memorize 4 classes and 4 ratios, no problem, go crazy, right? But if you can't, because you're not a memorizing type of person, you can always kind of reason your way through and understand that when we're having simple recessive epistasis, what that means is that one of the loci, when homozygous recessive, will mask the other. So now it doesn't matter that you had 3 RD and 1 RR. All of those become collapsed together into 4 that are essentially homozygous for E. Homozygous recessive for E, yellow labs. The next one that we'll discuss is duplicate recessive epistasis. So the thinking here now is either one as a recessive will mask the other. Now there is no one epistatic locus. Both are epistatic to each other. The example that we'll discuss, we have seen before in a slightly different context. We saw this little bit of a metabolic pathway when we talked about those Beetle and Tatum experiments, how they were playing around with mutants of this fungus, Neurospora crassa, and how they identify mutants for arginine, and they knew that because if they added arginine to the medium, now these things grow mold and whatnot. Cool. So now we know a little more about this pathway, and we know that there is ornithine, that's the original substrate in this segment of the pathway, I should say, that gets converted into citrulline by enzyme A. And then citrulline is converted into arginine by enzyme B. So it's a linear genetic pathway. What happens is, if and only if you have a functioning enzyme A and a functioning enzyme B, you will see arginine. Let's say that, I don't know, I don't want to confuse you with purple and white, which happens to coincide with the purple and white flowers of Mendel's. But let's say that we carry out some sort of biochemical test by which we can detect the presence of arginine through a chemical reaction that produces a purple pigment, right? So we would see purple pigment, we would see arginine only when both enzymes A and B are functioning. And when are enzymes A and B functioning? When their corresponding loci are anything but homozygote recessives. Right? So when the A locus is homozygote dominant or heterozygote, enzyme A is there. When the B locus is homozygote dominant or heterozygote, enzyme B is there. Again, we're dealing with full dominance, totally complete expressivity and penetrance and so forth, right? So heterozygote is the same as a homozygote dominant. The enzymes are there. When you have a homozygote recessive for the A locus or a homozygote recessive for the B locus, you won't see arginine. You won't see any purple pigment in your biochemical reaction. Why? In the first case, you're missing your enzyme A. That would apply to anywhere where there is a homozygote recessive for A. Second case, you might be missing enzyme B. That applies to all of those where there was a homozygote recessive B. And then, of course, there is a very special case of both of them are missing, which happens in that last cell over there, right? But in any of these scenarios, you're missing one, you're missing the other, or you're missing both, you can't get to arginine. When enzyme A is missing, ornithine cannot be turned into citrulline, and no matter how much enzyme B there is, enzyme B doesn't have anything to work with. And enzyme A may be present and producing kilos of citrulline in these cells, but if enzyme B is not there, it doesn't matter. You're not going to get any arginine because enzyme B is not there to make it. Did that make sense? So what happens here is that now, in a way, the double recessive or a recessive for either one, they all look the same. You're not going to get that phenotype, that readout at the end. Missing both or missing either of them, they all look the same. So all these three categories collapse into one. So instead of having a 9-3-3-1, you get a 9-7. Proportions-wise, remember, guys, that we say 9-7. If you're thinking of these in terms of fractions or percentages, what we really, really mean is 9/16 to 7/16, right? So if you're trying to map this to actual percentages or proportions of progeny, you have to remember that this doesn't necessarily mean that you will always get 16 individuals, 9 of which show purple and 7 of which show white. It's proportions. So if you're getting, I don't know, 57 individuals in the progeny, you would have to multiply 57 times 9/16 to see how many would be purple, and 57 times 7/16 to see how many would be white. Don't forget that. Just like when we say a 3-to-1 ratio, what we really mean is 3/4 or 3/4 and 1/4. And you can turn that into decimals if you win, so it's 0.75 and 0.25. So we're dealing with two loci. Of course, there can be epistasis with more than two, but we're going to keep it simple, so it's always going to be two loci. We went through simple recessive and double or duplicate recessive. What do you think comes next? Simple dominant, let's see. Yeah. Simple dominant. This is, in my opinion, the weirdest, right? So the scenario we're presented with is there is an enzyme B coded by locus B, and all this is, I think, very pedagogical. So it's for teaching purposes. I don't honestly think that the loci coding for these enzymes are necessarily called A and B. It's all for teaching purposes, right? But anyway, an enzyme B that converts a green pigment into a yellow pigment. Not much different from that. What was it? Dopachrome being converted into eumelanin. Then you have an enzyme A that no matter what pigment you have, it will destroy it. It will just degrade it. So you have no pigment now. Okay? So that's the scenario. What that means is that any time that enzyme A is there, and that means either homozygote dominance for A or heterozygotes of any kind, we indicate that as A underscore, meaning we don't know what the second allele is. It doesn't matter because this is a fully dominant trait. Right? So anytime that there is an A underscore, anytime there is a function, you won't see any pigment. In other words, you have no idea when A is functioning, you have no idea what the state of the B locus is. You have no way to know if the B locus was there working or not, and if these squashes were producing yellow or green pigment, because A took care of it, destroyed it all. And what you end up with is these white squashes. Only when A is a homozygote recessive mutant is that whatever was happening with the B locus will reveal itself. And that happens in this bottom quarter of the Punnett square, where you'll notice that in all of these cells, A is homozygote recessive. So now, all of a sudden, you can see what the B locus was up to. In the homozygote dominant or either heterozygote, yellow pigment. Only in the homozygote recessive for B would you see the green pigment. This picture here doesn't really do a lot of, doesn't do the whole math behind it a favor, because there is just as many white squashes as there is of the other colors. So if this is happening in real life, you would see very few green squashes, some more, but not many yellow, and the majority of them would be white. I didn't go through this. Let me do that real quick. So what's happening here is there is one of the dominants, let's say that one, that is, oh, there's a mistake here. Oh yeah, no, there's not. So one of the dominants that is masking the phenotypic manifestation of the other locus. So because it is a simple dominant epistasis, one of the loci, when dominant, will cover whatever you would expect from the other locus being recessive. So these two categories collapse into one. And last, as I overheard over here, yeah, double dominant or duplicate dominant. And this one goes also by a very special name that is called genetic redundancy. And the idea here is this is very common. This is something that has very high adaptive value. So this is something that happens often throughout evolution. And that is that there is more than one way to get to a final destination, right? We call it genetic redundancy because there is several genes or genetic pathways that do the same. So if one fails, there is another one as a backup or safeguard mechanism. That's why genetic redundancy is relatively common, right? So in this particular example, we have a species of rice in which there is a yellow substrate that gets converted into a brown product, a brown pigment, by either enzyme A or enzyme B. Okay? So now, all you need is either of them working, and you'll see that brown product. So the easiest way to really understand this is to flip it around and say, okay, when will I not see the brown pigment? When will I not see brown rice grains? The only time you're not going to see brown pigment, brown grains, is when both fail. And the only cell in the square in which both fail is the double recessive. Everywhere else in the square, you'll have either one or both of the enzymes working, and all the yellow substrate will turn into brown product, and those rice grains will be brown. So all of these in which there was either or both of the loci with a dominant in them are indistinguishable from one another. They mask each other or the absence of each other in a way, and they all collapse, the 9-3-3 all collapse into a 15. So as I was saying before, there's two ways to learn epistasis. One is to match simple recessive epistasis. It's 9-3-4. Duplicate recessive epistasis, you go down the list. The other one is to kind of remember this, that in a simple recessive, there's going to be one of the recessive masking the dominant of the other, simple dominant, one of the dominants masking the recessive of the other, and so forth, and then collapse the numbers accordingly. The trickiest bit is to be able to recognize each of these different types of epistasis from maps like this. That's possibly the trickiest bit, where you have to get out of the Punnett square and get out of genetics and try and understand from a process point of view what's going on, and how if you have a yellow substrate that can be converted into brown product by either enzyme, so then what that means is that the presence of either will give you the phenotype. Does that make sense? That can be slightly trickier. And no better example for that than the Labrador dogs and the squashes. If you go back to those slides, you'll notice that the map is very similar. There is a vertical reaction here controlled by one locus and a horizontal reaction controlled by the other. The difference is that the horizontal reaction was not taking place when one of the loci was homozygous recessive, whereas the other horizontal reaction was always taking place anytime that the second locus was dominant or heterozygote. Just takes a little practice in either way of memorizing what's going on with these deviations from the 9331 into rather predictable distributions. So a double dominant or duplicate dominant will always be a 15-1, a single dominant will always be 12-3-1, and so forth. This last slide for your entertainment later, basically to show all of this in action. So here in this table, which is from your book, there is the different traits that can contribute to a cat's fur, the different phenotypes associated, or that first column should be character, by the way, because fur length is a character, not a trait, but phenotypes, and then the genotypes. And what this shows is that some of them show incomplete dominance, some of them show interactions with other genes, some of them show sex linkage. So when it comes to predicting the fur of a cat, we can. It's just that it's going to take several steps because we need to figure out what aspects of it are going to be sex-linked, what aspects of it may have some epistasis going on, what aspects of it may follow simpler Mendelian genetics, but with confounders like incomplete dominance and stuff like that.